DNA Replication by Liv and Brenna. Each individual nucleotide is built and makes one side of the DNA strand. This strand is built from 3 to 5. Next, the opposite strand is being built, which goes from 5 to 3. This strand is put together, and then the two strands are put together and sealed by hydrogen bonds. Anti-parallelism, because they are going in the opposite direction, although they are parallel. Now they are being put together by hydrogen bonds. To replicate the DNA, helicase comes into play, which is an enzyme. Primase is an enzyme that puts RNA primer to attach onto the replicated strand, or to make the replicated strand. Because thymine isn't present in RNA, it is replaced with uracil. On the leading strand, the primer allows the replicated DNA to continuously grow, but on the lagging strand, multiple spots of RNA primer need to be added on in order to build, because it only builds from 5 to 3.
DNA Replication by John Riven. It's a blue, I said yellow.
Okay, really?
Come on, John.
These RNA primers are also attached to the DNA by hydrogen bonds. An enzyme called DNA polymerase connects by hydrogen bonds from the primer, and these DNA nucleotides were built individually before being added onto.
Because anyway, we
I was waiting for you to stop.
Helicase then moves further up the DNA strand and separates the hydrogen bonds. Once again, the primer is then added onto the other side, so the lagging strand. But since on the leading strand, it doesn't need to be added on anymore because of how it's being built from 5 to 3 prime. Nucleotides were built individually before being added on to the other DNA.
He looked at me and pointed. Primase adds an RNA primer that which the replicated strand of DNA can be built off of.
The primer was added on before the DNA.
Alright, John.
This is the leading strands because it originates from 5 prime and moves to 3 prime. It also only shows one primase. This is the lagging strand because it goes from the opposite direction, because it started in 3 prime and ended in 5 prime. So it had to be built in a different way, where primer was used multiple times. DNA polymerase 1 then separates the hydrogen bonds between the RNA nitrogen bases, or nucleotides, from the DNA ones, and replaces the backbone that is RNA with DNA. This is done on both strands. Each nucleotide is built again separately and then added together again by hydrogen bonds. Because on the lagging strand there are multiple cases where the primase was used, there's multiple cases where DNA polymerase 1 has to break down the RNA backbones. And because RNA is being replaced with DNA, thymine can be placed back into the picture.
Alex, what is that? Alex, what is that? Is that a yellow or a purple? Where? Did this go better in your
And this is done again at the top of the strand.
Wow, man, what a good idea. So,
Ligase is the last enzyme that is used, and it seals any gaps that are in between each nucleotide or each piece. And finally, the two DNA strands are identical because of DNA replication. This was supposed to be the lagging strand, but is now identical to the leading strand. And